are talking about the principle of DNA electrophoresis techniques, the specialties for DNA nucleic separations, and these applications have changed quite a bit in the last 2 decades. I will talk a little bit about what happened and what has changed, and where we stand today with the DNA electrophoresis techniques. So in the first slide, I just want to a little bit to remind you on how the principle of electrophoresis is working. So you have charged particles, charged molecules. In the case of nucleic acids, you have only one possibility to charge them negatively. You usually use a basic buffer system, and so if you introduce this into an electric field, the negatively charged particles will start to migrate in the electric field towards the anode. And different types of components have different electrophoretic mobilities. They have a different speed. There is a very simple equation here which you can easily remember. It's not the official one for electrophoresis, but it's a equation which is easy to understand and easy to remember, and that just takes you shows you the relation between the velocity of a particle molecule and the electrophoretic mobility. So the velocity is the product of the electrophoretic mobility, a quite complex unit, times the electric field strength. The electric field strength is the driving force. It's measured in volts per centimeter. So it's the local force which moves the particles into the direction of the anode. And the mobility is quite complex. In the next slide I will show you how the mobilities are dependent, from what they are dependent. On the right side on the animation, you see that by these different migration speeds you produce different zones, so you get a separation. You can do that in a gel, for instance, and then you can stain the gel and find the zones afterwards. So how are the dependencies of the electrophoretic mobility? First of all, it's the gel pore sizes. So this is a parameter which is active on all particles, on all components, because if you have a gel with small pore sizes, generally the electrophoretic mobilities will be lower. If you have a gel with more open structure with bigger pore sizes, the electrophoretic mobilities in total will be higher. And then there is also the buffer which acts on all the particles. For the pH value, the ionic strength and the conductivity, which is connected, is responsible for the mobility, and the viscosity. Just an example, you can modify the viscosity of a gel by not changing the pore sizes, but just the viscosity. You can slow down the migration, the electrophoretic mobility, just for instance by adding a dextrane to agarose gel. You can slow down the mobility but not change the pore sizes of the gel. And the temperature is also very important. The higher the temperature, the higher is the mobility. And in DNA sequencing, where you work under denaturing conditions, in the past time when gels were used, there was very often the problem that the temperature in the center of the gel was higher on the than on the two lateral sides. So people were using heated plates, gel plates, for running the sequencing fragments, and in this way achieving a straight front and better comparability of the band positions. These are the parameters which act on all the sample components, and then the sample components differ, for instance by charge, The more charges, the more negative charges you have on a molecule, the faster it will run. But in the nucleic acid electrophoresis, the most important is the size of the nucleic acid. So the smaller ones migrate faster than the larger ones because of the sieving properties of the gel. Another important thing you should always remember are the electric parameters. So the voltage which you apply on such a gel is dependent on the separation distance, on the length of the gel, the distance between the electrodes. So in order to achieve a certain volt per centimeter field strength, you need a higher voltage for a longer gel than for a smaller gel. For instance, a sequencing gel with like 40 centimeter long gel will need about 4 times as much voltage than a mini gel with 10 centimeters. So that is one part. And the other is important to know that the conductivity, the current which goes through a gel is dependent on the thickness times the width, the cross-section of the gel. So if you, what you can, run several gels with one power supply, you have to multiply the current setting, the maximum current setting. If you run 3 gels, you need to multiply the current setting by 3. But don't modify the voltage. And the power is the product of voltage times the current. And the power is responsible for producing the heat. So the temperature produced in the gel is higher if you have a higher power placed set on a gel. Okay. Now, another important thing to know is what is electroendosmosis and what is the influence of electroendosmosis. It's a short name EEO. So I want to explain you shortly what is electroendosmosis. On the left side, you see the electrophoretic migration. So the yellow thing they see here should be a gel matrix, agarose gel or polyacrylamide gel. And here you see the electrophoretic migration. The smaller particles, negatively charged, migrate faster than the bigger ones towards the anodal direction. Okay, easy to understand. But very often the matrices are not completely inert. We know that very well from agarose gels because agarose is a natural product produced from red agar seaweed, and from this red seaweed, and it is produced by removing agaropectin. The agaropectin contains carboxylic and sulfonic groups which disturb a little bit the separations. So by purifying it, you try to remove completely these buffering groups, but this is not possible. So there will always be some remains of carboxylic and sulfonic groups in agarose gels. And another point here is if you are working on glass or in glass tubes or in glass plates, also the glass surface is not completely inert. It contains silicium oxide groups. So when you use a basic buffer system, which is always used in nucleic acid separations, these buffering groups, carboxylic groups, sulfonic groups, silicium oxide groups become deprotonated. That means they become negatively charged and attracted by the anode. So they would like to migrate towards the anode, but they can't because they are part of the matrix. So they cannot move. That means that there will be a counter reaction. And this counter reaction is a migration of protonated water into the cathodal direction. This is truly a water flow. It's called electroosmotic flow because of these charged groups. Now, different agarose materials, different agarose qualities have different electroendosmosis values. So the cheapest ones have more of these charged groups than the very expensive agaroses. And you can find the definitions when you try to buy agarose. You will see a value which is called -mR or sometimes also signed with EEO, the electroendosmosis value. In the electroendosmosis value, the higher it is, the worse is the quality, the more influence of the electroendosmosis can happen. What is negative with the electroendosmosis? It migrates into the opposite direction than the electrophoretic migration, so it can cause disturbed bands and unsharp bands, blurred bands. And in agarose gels, very important, it can dry out the anode and flood the cathode. That's why the boxes for agarose electrophoresis for nucleic acid separations are so-called submarine boxes, where you have the gel submerged below the buffer surface. So the water flow, you cannot see the water flow because the water flows towards the cathode but will reflow to the anode because it's all under water. The electroendosmosis definition is tested by applying a mixture of dextrane, which is neutral, electrophoretically neutral, and albumin on a gel. And then after a certain time of migration, the distance, the migration distance of the dextrane is divided by the migration distance of the dextrane plus the migration distance of the albumin. And so you will get this thing. The dextrane usually electrophoretically would not migrate at all, but with the water flow it will be moved towards the cathode. And also the albumin will run shorter, in a shorter distance, the higher the electroendosmosis of the gel. Okay, just generally spoken, good agarose is expensive, and the best agaroses have the lowest electroendosmosis values. Here I have a comparison between agarose and polyacrylamide. So agarose basically you buy as a powder, you weigh it in the buffer, you heat it together with the buffer, and then the agarose gets dissolved, and then you chill it down, you cast a gel by just spreading the solution on a horizontal plate. Usually it's a plastic plate, and then you chill it down, you wait a few hours, then you will have a final building of the matrix. The matrix is very special in agarose, so you have a multiple helix walls around the pore size built by the agarose sugar molecules. That is the reason for good mechanical strengths with very large pore sizes. So you see a 1% agarose gel will produce about 150 nanometer pore sizes, and some people worked even with very diluted agarose gels, which are of course mechanically not very stable, but they were able to separate viruses, entire viruses particles with a 500 nanometer open gel. The polyacrylamide gels are made in a completely different way. You are using a radical polymerization of acrylamide monomers and a dimer, a cross-linker, which is usually methylene bisacrylamide. And by adding some starters, you will get a chain reaction and produce a three-dimensional gel matrix, which is defined by two values: by the total acrylamide concentration, so you are adding acrylamide plus bisacrylamide, the cross-linker, divided per volume times 100, so you have the volume for the gel. So the higher the T value, the smaller are the pore sizes. And you need to define it by a second parameter, which is the C value, the cross-linking factor, which is in most of the cases defined like this, as a relative value, gram bisacrylamide divided by the sum of acrylamide plus bisacrylamide. Please note that not everybody is defining it in the same way. So you find in some molecular biology labs another definition, the C value is the gram acrylamide in the liquid percentage, mixed percentage. You have to find out which is the value that you are working with, because if you mix that up, you can get different gel properties. Here I show an example for a very big pore size. For a polyacrylamide gel, it's a low concentrated gel to have 5% T and 3% cross-linking, and then you will get a 5 nanometer gel. This is a low concentrated polyacrylamide gel. Now compare that 5 nanometers with the agarose. There you see already is a big difference between the properties of a polyacrylamide and agarose gels, and so are the applications. So for larger nucleic acid fragments, you are using agarose for separations. For small kilo base fragments, for some amplification, PCR amplification fragments which are short, you are using polyacrylamide gels. I will come back to this in a moment. First, to agarose gels. So this is usually how you run them. You cast the agarose gel in such a tray, and you use a comb to produce the sample wells for sample application. The sample usually is you add some glycerol or sugar to make it a little bit more heavy than the buffer, in order that it does not mix with the buffer. So you can just pipette it into these little pockets, and you run the separation under the fluid level. So this is so-called submarine technique. On the right-hand side, you see a typical result. Detection of DNA fragments here is mostly used with fluorescent dyes. Formerly people used exclusively ethidium bromide, but as we all know, it's not a nice stuff. It's intercalating in the DNA and also in the human DNA, of course, so it's highly toxic, and people like to have something else instead. So you are evaluating these bands under UV light. There you see them shining out of the gel. I already said that there are different types of agarose gels. So here I show you a selection of the agarose gels that SERVA is offering, quite many different. Low melting is sometimes needed for special applications. I will show you. And so you have to, the best is to choose the agarose according to your needs. So here you see which agarose we recommend for which application. For routine analysis, the normal SERVA agarose. For PCR, special ones. For blotting, almost for the southern blotting, you can use any you want. But for recovery of DNA or RNA, a low melting agarose is easier, and also for in-gel reactions. Okay. Now here you see some examples of the chambers that are offered by SERVA, from the very small one, the Blue Marine 100, with a 7 × 10 centimeter gel format, to the larger ones, the Blue Marine or Blue Marine HTS. Here you can run longer separation distances, or if you wish, if you have more samples, you can run a higher number of samples just by forming different rows, arrays of pockets by using several combs. So you can run them in short distance in different positions. And with the red color here, you can see the marker for easier applying the samples. The same is the case for the Blue Marine HTS. Here you see detection example with the SERVA DNA stain clear G. So this is a not intercalating dye, and it's so far it's therefore not carcinogenic, and the reason is it does not intercalate. It sticks to the phosphate groups of the molecules. So that's the way it works. Now, for the illumination, at UV, you can either use a UV table, like shown on the left side, or better, you have a box where you are not exposing yourself to the UV light, and with an inbuilt camera system you can take the images much easier. This SERVA DIAS is already the third generation of it, includes also a 1D analysis software, which is also has been launched very shortly. It's a very well developed, new, easy to use evaluation software. In order to make it easy to interpolate the sizes of the fragments, you can co-run DNA standards. Here I show you 3 examples which are available from SERVA, and you will apply this one which fits best to the nucleic acid sizes you are looking for. Mostly the people are using tris-borate EDTA buffers for nucleic acid separations. That's the standard. You have the TPE buffer in the gel as well as in the buffer reservoirs. But in some cases people prefer to run a TPAE buffer, which is a tris-acetate EDTA buffer. The TPAE is I forgot to say, tris-borate EDTA buffer. So particularly when you run very high base pair DNA, maybe TAE is preferred. And third, this is coming more done in polyacrylamide gels, which I will explain a little bit later. We found out, we could see that the tris-acetate, tris-trisine, this is a discontinuous buffer system, works very well for separating short DNA fragments. So here you have the tris-acetate in the gel, and you have the tris-trisine in the running buffer, which gives a discontinuous gel effect, which works very well for small molecules. The most applications of agarose gel electrophoresis is restriction enzyme digest analysis, then PCR analysis, isolation of fragments for cloning and others, and RNA analysis and hybridization with Southern blotting. With Southern blotting, you are transferring the separated bands with the help of capillary forces onto a nitrocellulose membrane, and then you can perform hybridization with the DNA you are looking for. A special modification of agarose gel electrophoresis is the so-called pulsedfield gel electrophoresis, and that is needed for DNA molecules which are larger than 20 kilobases, because above 20 kilobases the DNA molecules are not separating anymore. They will migrate in one band, because then the migration is performed in a reptation migration. So the DNA molecules orient themselves in the same direction, and then they run by reptation in the same direction. Now, when you force these DNA molecules to change the direction by changing the direction of the electric field, the larger molecules need a longer time to redirect into this next direction than the shorter ones, and that gives you a very good separation. There are different systems on the market. One very famous is the hexagonal system, that you see here, but there are also other, like turntables and turn tablets and turning electrode systems, or here you see a field inversion gel electrophoresis where you just switch into the two directions. It is necessary to have an angle of at least 110 degrees, otherwise the rotation direction will not change, and then you won't get a separation. The pulse times are also dependent on the sizes of these DNA molecules. So here at the right bottom you see an example of chromosomal DNA run in such a gel with long pulse times. So such a separation can take several days if you need. Polyacrylamide gels are used usually when you run PCR products which are relatively short, like between 50 and 1500 base pairs, because here the resolution of the agarose is no longer as expected. Also, when you increase the agarose content, it will not improve it too much. And the sensitivity of ethidium bromide in DNA is also at the limits. So in such a polyacrylamide gel, you can apply either silver staining or fluorescent labeling, and then you can get down to a 15 picogram per ven sensitivity. But note one thing. In polyacrylamide gels, when you run them under native conditions, if you run them under denaturing conditions, high temperature in presence of urea, the migration is absolutely according to the sizes. But when you run them under native conditions, A and T rich fragments migrate slower than C and G rich fragments, and so you get also you don't get direct separation according to the size. One example you can directly observe is when you use a ladder which has a spike in order to see that the spike is usually at about 800 base pairs. But in a native gel it will shift to 1500 base pairs. The reason why this is the case is polyacrylamide gels are more hydrophobic than agarose gels, and so you can have interactions between the AT rich fragments and the matrix, and the C and G rich fragments have much less interaction with the gel matrix. Here you see also a lately developed system for polyacrylamide gels run on a horizontal plane. So these gels are not run under buffer because they have very low electroendosmosis. Here you see a very spectacular separation with 103 samples for viewing PCR products. These chambers have been developed for DNA page, but also for protein polyacrylamide gel electrophoresis and isoelectric focusing work very well on a benchtop. And there is a smaller system available for vertical separations also. Very easy to handle, so you can slide in ready-made gel cassettes and clamp them to the core of the chamber and get very nice separations. Here is one example, that is the SERVA gel prime. This is a series of gels with different concentrations. Here you see one example with 14% T value, and these gels contain a tris glycine chloride buffer, but they are run with a TBE buffer in the running compartments, in the buffer compartments, and also the samples are applied in the TBE buffer. And here you see, after staining the gel with DNA stain clear G, a very super nice result in such a gel which is only 8 × 8 centimeter large. So here just a view into the past. So DNA sequencing started in vertical, long, very, very thin gels with radioactivity labeled as PCR products or fragments, DNA fragments. Then people found out it's much better to do an online detection by labeling the reactions with fluorescent tags, with different fluorescent tags, and running them in such a gel with a direct detection. This was a long time done in these gels until the capillary technology came up, and with the capillary it's even easier to run many samples. So the biggest machines that were running 386 sample well and capillaries at the same time, reactions at the same time, and so the human genome, for instance, when it was detected, that was done with such a multi-capillary electrophoresis system. At present, these systems are still used a lot for DNA cloning controls and SNP, single nuclear polymer analysis. No, polymer, sorry, the word is not coming in the moment. Polymorphism analysis. And DNA sequencing nowadays is done by non-electrophoretic techniques, the so-called next-generation sequencing methods, which have very, very high throughput and need only very low amounts of material. Yeah, that was a short overview on DNA electrophoresis techniques, and I'm wait for your questions. Thank you for listening.